Okay, continuing. There are few structures which are really important. So from lateral ventricle to the third ventricle, the cerebrospinal fluid, it travels via the foramen. And which foramen is it? Foramen of Monro. So foramen of Monro, very important. And foramen is singular, but there are two of them, right? So we are gonna say foramina, which is plural, right? And this is singular. So foramen of Monro or foramina of Monro, right? So it allows the CSF to travel from lateral ventricles to the third ventricle. Then after this, you have a cerebral aqueduct, which is also known as aqueduct or Sylvius. It's also known as aqueduct of Sylvius. So cerebral aqueduct that allows the transfer of cerebrospinal fluid from the third ventricle to the fourth ventricle. And here you have foramina of Luschka on the lateral sides. So this is also known as the lateral aperture or foramina of Luschka. So you can remember this Luschka. You can remember this by L and L, lateral and Luschka. And right in the center, medially, you have foramen of Megendie. How do you spell it? Magendie, Megendie, or whatever you want to say it. So this is medial. So yes, these are very important structures that you should be familiar with. Starting off, first of all, sorry. So you have foramina of Monro, then you have aqueduct of Sylvius, then you have foramen of Luschka at the side and foramen of Megendie in the middle. So these are the foramina. Now, how does the cerebrospinal fluid, how is it made? So let's dive deep into that. So basically, what happens is that you all must be knowing that this is the gray matter. Sorry, this is the brain matter, right? So this is brain matter. And right outside the brain matter, which covering do you have? Which meningeal covering do you have? You have your pia mater, right? So you have the pia mater, which is the innermost meningeal layer, pia mater here, right there. So this is pia mater. Good. And what is this? This is your brain, the main matter of the brain, right? And in the middle, you will find what? You're going to find the cavity. This is the ventricle in the middle. This is the ventricle, right? In the middle. And even more important, you will notice that this ventricle is lined inside with the help of ependymal cells. These are the ependymal cells that line the ventricle. So, am I making sense? Let me just label this as well. This is ependymal cells. This is ependymal cells. And then here, this is your pia mater, brain, and then ependymal cells. Now, what happens is that here you have your choroidal arteries and choroidal veins. So the choroidal artery is coming in, it's forming a tuft here, and then it's going out. So vein comes in, sorry, artery comes in, and the vein goes out. So it's basically a tuft of the vascular structure. And along with this vascular structure, there's one more thing that's being pulled inwards, and which is the pia mater. So pia mater is also being pulled inwards with this choroidal artery. And not just choroidal, not just pia mater, but okay, before that, let me make it clear that this pia mater and that choroidal artery, they together make this very important structure called tela choroidi. What is the structure? Tela choroidi. Yes. And when this tela choroidi, okay, so not just tela choroidi, but there's one more thing as well. What is that? That is these ependymal cells, they start growing here as well. So tela choroidi plus ependymal cells, these together are known as what? As choroidal plexus. So now I'm sure you can understand that what is choroidal plexus. So pia mater plus the choroidal artery, they together make tela choroidi, and tela choroidi plus the ependymal cells, they together make the choroidal plexus. So I hope this is clear that first you have the choroidal artery and pia mater, tela choroidea and tela choroidea plus your ependymal cells, that makes the choroidal plexus. Now, what's the function of choroidal plexus? So they have the main role of producing the CSF. So how is CSF made? It's stored in the ventricles, but how is it made? It's made by the choroidal plexus. And how exactly does it do that? So let me tell you that this, for example, if this is your choroidal plexus, right? So obviously it will be lined by a lot of cells, epithelial cells or whatsoever, right? So here it has channels also, and those channels do what? Sodium is excreted out into this ventricle, into the cavity, right? Sodium is pumped outside. So actively sodium is pumped out, and calcium follows along passively. So active transport for this and diffusion or passive transport for chloride. And because sodium and chloride both went out, water also diffuses out. And how about glucose? Glucose is also carried out, but in very less quantity compared to the glucose concentration in your blood plasma. And what about potassium? Potassium goes inwards. So potassium is one thing that goes inwards or in the opposite direction. So these are all the constituents of your CSF. So this is how the CSF is made. I hope that's clear. So this is how CSF is made. How is it made? Again, active secretion of sodium. Passively, it is followed by chloride and water, and then glucose also. And how about potassium? Potassium is reuptaken or it's reabsorbed into the uh, capillary or artery or vein or whatsoever, right? So these are the constituents of your CSF. So choroidal plexus. What is choroidal plexus? Tela choroidea plus ependymal cells. And what is tela choroidea? Tela choroidea, it's the blood vessel, which is the choroidal artery and the pia mater, right? So these all structures collectively, they make the choroidal plexus, which helps in the production of the CSF. And where is CSF produced? It's where is it stored? It's stored in the ventricles, the fluid-filled cavities. Right, and how many fluid-filled cavities we have studied? There are two lateral, one third ventricle, which is between the thalami in the diencephalon, and one is the fourth ventricle, which is in the rhomencephalon, and lateral is in the telencephalon. Right. So yes, that being said, now very important. Let's move forward. Now we are going to talk about. Let's add some things here. So where's subarachnoid space? Where can you see the subarachnoid space? So let me label that. Here you can see I haven't labeled it properly, but this is your pia matter right outside the brain, and then this is the dura matter. Sorry, arachnoid matter. So between pia matter and arachnoid matter, the entire space, this right, this entire region is the subarachnoid space. This is the subarachnoid space, and this is where you're going to find the CSF. Now, one more layer should be there, the third meningeal layer, which is, let me draw that as well. That layer is, let me choose a good color, um, dura matter. So let's draw dura matter. Dura matter is there everywhere, right, like this. And now, dura matter has two parts. It has an outer layer and an inner layer. The outer layer is the periosteal layer, which is closer to the brain, to the bone, sorry, and the inner layer is the meningeal layer. So, meningeal layer is closer to the brain. So, yes, meningeal layer closer to the meninges. And then there's a periosteal layer. This separate and this separation can clearly be seen in the topmost part here, which is the superior sagittal sinus, superior sagittal sinus, superior sagittal sinus, sup- also known as the dural sinus, dural venous sinus, also known as the dural venous sinus. Now, what happens exactly is, so this is that cavity, right? How did I label it as? Superior sagittal sinus or dural venous sinus, right? So it is between the periosteal and meningeal layers of the dura mater. So this is the dura mater. And what is this? This is the subarachnoid space. So subarachnoid space, and this is the superior sagittal sinus. I hope that is clear. Now this is one structure that you should know, and let me show that to you here as well. Here you can see this is fox cerebri. What is that? I'll tell you in a second. That's a layer of your dura mater, and this is your arachnoid matter, which is the pia mater, the innermost. Then you have arachnoid matter. Between the pia mater and arachnoid matter, you have the subarachnoidal space. So this is pia mater, this is arachnoid matter. So you have subarachnoidal space where your CSF flows. And then above the arachnoid matter, you have the dura mater. Dura mater has periosteal layer and a meningeal layer. So periosteal layer and meningeal layer, they separate here. That is the superior sagittal sinus, or you can also call it the dural venous sinus. Right here there is a separation, and the space between, I told you pia mater and arachnoid is subarachnoid, but the space between the arachnoid and dura is subdural. So subdural and subarachnoid space. Okay, I hope that's clear. So this is the image which makes it clear. And yeah, I was talking about fox cerebri and all of that. So let's just clear that up quickly. And again, here you can see this is the superior sagittal sinus, this is the inferior sagittal sinus, and here you can see the cavernous sinus. In this diagram, you can see the cavernous sinus better. This is the cavernous sinus, and here you can see the superior sagittal sinus. This is the inferior sagittal sinus, and this is the tentorium cerebelli, which is placed horizontally, and this is the fox cerebri, which is placed vertically. So fox cerebri and tentorium cerebelli. So these should make something clear. Again, what is this? This is fox cerebri, which is placed vertically. Fox cerebri, and here you'll see tentorium cerebelli, which will be horizontally. What is fox cerebri and tentorium cerebelli? They both are folds of dura mater. They both are folds of dura mater. So remember, dura is the outermost layer, right, of the meninges. So this is fox cerebri and this is tentorium cerebelli. So fox cerebri is a sickle-shaped fold of dura mater that separates the two cerebral hemispheres of the brain. So it is placed between the two cerebral hemispheres of the brain, as the name suggests. It runs in the longitudinal fissure. It provides structural support to prevent the brain from excessive movement and helps prevent certain types of brain herniation. Attaches from the crista galli of the front to the occipital bone at the back and contains the superior and inferior sagittal sinuses. So this contains the superior and inferior sagittal sinuses. Right. And how about tentorium cerebelli? It is a tent-shaped fold of dura mater that separates the cerebrum from the cerebellum. So it separates cerebrum and the cerebellum. So it's placed just above the cerebellum. So if you can see this diagram that I've drawn, so this is your cerebrum and this is the cerebellum. So this is a tent. So tents are normally like this, right? So this is a tent for cerebellum. So it separates the cerebellum from the cerebrum. So yes, its primary functions are to support the weight of the cerebral hemisphere and protect the brain from trauma. So it supports the weight and it protects the brain from trauma. It divides the cranial cavity into the supratentorial and infratentorial spaces and has a free anterior edge with a notch called the incisura tentoria, which allows the midbrain to pass through. Right. So this was extra information. Now moving forward, very important, ventricles. Let's come back to the ventricles. First, let me just clear the anatomy of ventricles, and then we'll talk about the circulation of the CSF. Here you can see this is your lateral ventricle. This entire structure is the lateral ventricle. Then where's the third ventricle? So first, clearly you need to know that this is the lateral ventricle, this entire thing. This is its this is its anterior horn, this is the body, this is the inferior horn, and this is the posterior horn. The posterior horn is also known as the occipital horn. The inferior horn is also known as the temporal horn. The interior horn, right? There's interior horn and body. Body is in the parietal lobe, and interior horn is in the frontal lobe. So frontal horn, and this is the temporal horn. Body is basically in the parietal lobe, and posterior horn is the occipital horn. So this entire structure is the lateral ventricle. So you need to know the interior horn, posterior horn, inferior horn, and then the body. This is occipital, temporal, this is parietal, and this is frontal. Good. Then what about this structure? This is the third ventricle. So lateral ventricle, third ventricle. And where is the fourth ventricle? This is the fourth ventricle. So lateral, third, and fourth. Very important. And the what connects the lateral with the third ventricle? What was that? Those were the foramina of Monro. So these are interventricular foramen of Monro that connect the two. And then what about the third ventricle with the fourth ventricle? That was cerebral aqueduct, also known as the aqueduct of Sylvius. So cerebral aqueduct, this thin canal right here. Then you had the fourth ventricle, and below that you had the central canal, right? So these are the structures. Then again, you can see in this diagram that this is the cerebellum, this is your pons, and then you'll have the medulla, and here you'll have the fourth ventricle, this region, fourth ventricle, and you can see the choroidal plexus. This is the choroidal plexus, right? And this is your central canal. And very important, the there's another very important structure that I want you to be familiar with. So let's once again, go over the anatomy. This is this entire thing is your lateral ventricle. This entire thing, lateral ventricle. This is the frontal anterior horn, this one, right? This is the central part. Then this is the temporal inferior horn. This is the occipital posterior. So anterior, posterior, inferior, and body. So this is the lateral. What about this is right lateral and this is the left lateral. Then if you talk about this, the connection between the lateral and the third ventricle. What is this? This is the third ventricle. So third ventricle is this one. And the thing that connects both of these are left interventricular foramen of Monro. What about this? This is interthalamic adhesion. You normally get models for this in your OSPE, so it's super important for you to know. This is interthalamic adhesion. And then after that, right here you have Sylvius, aqueduct of Sylvius or cerebral aqueduct, which connects it to the fourth ventricle. And here in the fourth ventricle, you'll see you'll have foramen of Lushk on the sides, and you'll have foramen of Megendie in the middle. So the sides, lateral aperture foramen of Lashka at the sides here. There'll be two of them. And right in the center, you will have the median aperture foramen of Megendie in the center, right? So these are lateral apertures foramen of Lashka, and in the middle you have Megendie, right? And then this is another diagram that I don't want to discuss right now. And again, so this is superior sagittal sinus. This is inferior sagittal sinus, should be right here also. And then the posterior horn, we can see what else is there. These are the choroidal plexus that I can already tell. And what is the choroidal plexus of third ventricle? Okay, cool. And then interventricular foramen of Monro. And is this something that we need to know? Nothing. Everything. Yeah, there are two very important things that you need to know. Yes, very important. Let me change the color of this. So you have very important structures. One, okay, superior sagittal sinus that you already know. But very important, the lateral aperture, which is the foramen of Lashka. Here you can see the right, sorry, here you can see right here, this region is very important. Here, this is prepontine cistern. Now, this is a part of the subarachnoid space. This is the subarachnoid space, the prepontine cistern. And here, you'll notice one more cistern, which is the posterior cerebellomedullary cistern, which is again a subarachnoid space. Here is where the CSF will flow. And then you can also see the median aperture foramen of Lashka. So let's recall. This is the median aperture foramen of Lashka. This is the lateral aperture. Sorry. Median aperture foramen of Megendie, lateral aperture foramen of Lashka. Here, the subarachnoid space, this one right here, posterior cerebellomedullary, between the cerebellum and the medulla. So posterior cerebellomedullary cistern, and this is prepontine cistern right in front of the pontine, or you can call it cerebellopontine cistern, right? So these are the cisterns that you need to know, which are part of the subarachnoid space. Now, when CSF comes out, it comes into this cerebellomedullary, and where this Megendie, Lashkov, sorry, foramen of Lashka and Megendie, it also comes into the prepontine cistern and the cerebellomedullary cistern. Now let's look at the directions in which your CSF flows. So cerebrospinal fluid, it is produced by the choroidal plexus that we already know, which are located in the lateral ventricles. They're also in the lateral ventricles. They're also in the fourth ventricle and the third ventricle, at the in the roof of these ventricles. CSF flows into the third from lateral ventricle. It flows into the third ventricle through the foramen of Monro that we know. And then from the third ventricle, it moves where the aqueduct of Sylvius to the fourth ventricle. So from lateral to the foramen of where the foramen of Monro to the third ventricle, and from third where the aqueduct of Sylvius to the fourth, and from fourth ventricle it exits into the subarachnoid space, particularly in your what are the two points that I mentioned of subarachnoid space? Prepontine cistern, and the other was the cerebellomedullary cistern. And by the cistern, this word you should know, it's subarachnoid space. And how does it enter these 2 points? It enters them with the help of these two very important apertures. One is a median aperture and the other is the lateral. So median is your foramen of Megendie and lateral is your foramina of Lashka. There are two of them, so foramina. And how does it? Once in subarachnoid space, the CSF circulates around the brain and down the spinal cord, acting as cushion. The fluid is reabsorbed into the venous blood circulation, primarily through the arachnoid villi in the superior sagittal sinus. Now, this is the flow chart that'll help you remember. So, choroid plexus, that's where CSF is made. Then it goes to lateral ventricles. From lateral ventricles, where the foramen of Monro, it goes into third ventricle. In third ventricle, additional CSF is produced and added because this also has its own choroidal plexus in its wall, in its roof. Then where the Sylvian aqueduct, it goes into fourth ventricle. In the fourth ventricle, again, additional CSF is added because this also has its own choroidal plexus. And then where the foramina of Lashka, there are two of them, so foramina of Lashka, and where the foramen of Megendie, it goes into the subarachnoid space in the prepontine cistern and the cistern of the what do you call it, cerebellomedullary cistern. And then from here it just circulates around the central canal and the brain, right? And then it goes to the superior sagittal sinus where it gets absorbed, superior sagittal sinus, and it gets absorbed with the arachnoid villi into the blood. So again, you can see here that that's the entire pathway, and I don't want to recall it, but yes, where the arachnoid granulations, it gets absorbed into the blood. And yes, arachnoid granulations, let's quickly discuss them. So here I told you these are the dural layers. One is the periosteal and other is the meningeal layer. Here, what happens is that your arachnoid layer, which is the arachnoid one, the middle one, right? So it should be drawn in purple. This layer, a tuft of capillaries is there, but also that is surrounded by your this very important layer called arachnoid layer, right? And from here, your CSF is reabsorbed into the venous sinuses, into the blood. So it goes into the blood. So yeah, that's about it. I hope this lecture didn't get very lengthy. I tried my best to cover the anatomy and cover the entire circulation procedure. And if there was any extra information, I wanted to cover that as well.